Hello students, welcome to the channel. In this video, we solve the fifth and last passage of the reading section of the May 2021 US SAT. This is a paired science passage, so let's begin. Passage 1 is adapted from Morton E. Allentoft, Recovering Samples for Ancient DNA Research, Guidelines for the Field Archaeologist by Antiquity Publications. Passage 2 was adapted from Sam Keen, Ancient DNA by Chemical Heritage Foundation. Passage 1. Working with ancient and degraded DNA is not without challenges. Most serious, perhaps, is the risk of contamination with contemporary DNA, which can easily swamp the small amount of authentic DNA in an ancient sample. This fact became painfully clear in the earlier years of ancient DNA research when a number of high-profile publications seemingly presented evidence of DNA from samples of extreme age, including 80 million-year-old dinosaur bones and greater than 120 million-year-old insects. We are now aware that these results reflected modern contaminants, in part because the rate of post-mortem fragmentation of DNA excludes the existence of DNA that old. In response to these claims, a set of ancient DNA rules was established to minimize the risk of contamination and to provide some means for downstream authentication of the results. And since then, numerous studies have identified and assessed contamination problems in ancient samples. So the first paragraph of this passage is concerned with ancient DNA which is recovered from fossils, from archaeological deposits, etc., and how there is the risk of the contamination of that DNA with modern DNA, the DNA of today. And so this passage is going to discuss the rules that have been established to prevent that contamination. In essence, however, most of these rules represent symptomatic treatment. If a sample is contaminated with modern DNA before entering the laboratory, for example, during excavation, it can be difficult to remove the contamination, and it can be impossible to distinguish between authentic ancient DNA and contaminants. This latter problem is particularly pertinent if the target DNA and the contaminant DNA are from the same species, as is often the case when research is conducted on ancient human material. So one of the main problems of contaminated DNA is the problem of separating the authentic ancient DNA from the contaminated modern DNA. And that becomes acute when human samples, ancient human samples are being worked with, because the excavators, modern humans, and the ancient humans are, of course, the same species. So it's difficult to distinguish between the two DNAs. In the era of high throughput sequencing, statistical tools based on DNA damage signatures are now available to assess the overall authenticity of bulk ancient DNA data. Considerable time and resources, however, are expended before the contamination can be identified. And if a sample has been contaminated decades or centuries ago, it may result in contaminant DNA that displays degradation damage patterns similar to that of true ancient DNA. Although strict laboratory exercises are often combined with bioinformatic analysis to respectively remove and identify DNA contamination, it would be extremely beneficial if the risk of contamination could be lowered in the first place. Okay, so this paragraph talks about the other issue with DNA contamination, which is the fact that modern DNA is DNA, that could have mixed with ancient DNA over the course of the past century, over the course of the past 50 years, past decade. And that DNA also shows signs of degradation, which are similar to the degradation observed in ancient DNA. And that compounds the problem of separating the authentic ancient DNA from the contaminated modern DNA. Okay, so let's answer questions pertaining to passage 1. In passage 1, the main purpose of the second paragraph, lines 21 to 31. So we are looking at these lines. What is the main purpose? it presents how most of these rules represent symptomatic treatment, which is to say that once contamination has already occurred, how do you get rid of it? It doesn't really, there is no prevention per se. No rules for prevention have been assigned per se, right? This latter problem is particularly pertinent if the target and contaminant DNA are from the same species. So the main purpose is to explain in greater detail the nature of newly established procedures mentioned in the preceding paragraph. Assert that a solution presented in the preceding paragraph is inadequate for addressing the full scope of a problem. Emphasize the necessity of using different research technologies depending on the species to which the ancient DNA belongs. Describe the different kinds of contaminants that researchers working with ancient DNA have discovered through recent studies. So it's B, right? Because we are saying here that we have come up with rules to minimize the risk of contamination, but even these rules are at best symptomatic treatment. And if you are even, if you come across a sample that's been contaminated, then it is impossible to decontaminate it. So that's a major problem. So this is kind of asserting that a solution that was presented, the rules, is not adequate for addressing the full scope of a problem. 44, it can reasonably be inferred that the attitude of the author of passage 1 towards the technology that has now become available for identifying and removing DNA contaminants is. So this is here, right? Considerable time and resources are expended before the contamination can be identified. And if a sample has been contaminated decades or centuries ago, it may result in contaminant DNA. So he acknowledges those technologies, but he says that they do require time and effort. Although strict laboratory exercises are often combined with bioinformatic analysis, it would be beneficial if the risk was lowered in the first place. So the author's attitude is somewhat cautious because the technology remains costly to use but does not guarantee accurate assessment of DNA. Clearly dismissive because the technology is narrowly limited in the type of DNA samples it can analyze. Genuinely hopeful because the technology resolves ongoing challenges but does not interfere with the study of DNA. Openly appreciative because the technology makes the work of analyzing ancient DNA considerably more straightforward. So I think C and D are very positive and B is very negative, right? I think A is a good answer because it clearly states here that considerable time and resources are expended if a sample has been contaminated. The other issue is that laboratory exercises can be used to do that. It would be beneficial if the risk of contamination. So he's cautious. He says that yes, these technologies are available, but they are costly and they do not guarantee the accurate assessment of DNA. Right? Right. So A is a good answer. Which choice provides the best evidence to the answer to the previous question? So I would say anything from 35 onwards. Yeah, so I'm going to look at C and D, 32 to 35 and 35 to 40. In the era, yeah, this is just describing that the technologies are present. How the author views them is in 35 onwards. So D. Okay, as used in line 41, strict most nearly means 41. Although strict laboratory exercises are often combined with bioinformatic analysis. So strict here would be rigorous, right? You need to be rigorous, you need to be careful, you need to be extremely meticulous when you're applying these exercises to separate contaminated DNA from authentic DNA, right? So it's not absolute, it's not grim, there's nothing sad about it, and not narrow. So rigorous. Okay, 47 is about passage 2. So let's read passage 2. Studying ancient DNA, DNA is a lot like playing whack-a-mole. Stamp out one problem and another will pop up and take its place. Contamination, corruption, chromosomal shredding. It's a miracle scientists can even find DNA in specimens, much less glean information from it. But a few recent breakthroughs have greatly expanded our ability to read DNA and have already opened whole new vistas of evolution. DNA disappears after cells die for a few reasons. All healthy cells have enzymes that shred DNA to recycle it, and unfortunately, those enzymes keep right on shredding after death. Water and oxygen in the environment also react with DNA's backbone and degrade it further. I would say these processes reduce DNA to confetti, but that doesn't do justice to just how thorough the destruction is. It's more like confetti making its own even tinier confetti, which in turn makes its own confetti. After a few thousand years, a multi-billion base pair genome might be reduced to scraps a few dozen letters long, 100 million fold reduction. So this paragraph kind of introduces the level of degradation that ancient DNA can undergo um, as it interacts with the environment. And the analogy and the way it's described, this level of degradation is intense. And the previous paragraph was talking about how finding ancient DNA and being able to get information from it is a hugely difficult task, but there is some new research that throws light on how this can be done. The best aDNA comes from samples unearthed in tundras or caves. Recent advances in computing power have also allowed scientists to assemble sequences from even minute scraps of DNA. The oldest recovered genome so far came from a horse's leg bone buried in Canada's Yukon Territory for 700,000 years. With the right sample and ever better software to analyze it, scientists think they can push that back to 1 million years. So this talks about the scientific advances that have made the study of aDNA feasible. Finding the right sample solves only one problem though. In most ancient tissues, 99% of the DNA present comes from contamination by invading fungi and bacteria. So scientists have to deploy chemical snares like RNA bait. If searching for human DNA, the researchers would prepare the bait by manufacturing millions of strips of human RNA, DNA's chemical cousin, albeit with one modification. These RNA strips have chemical Velcro attached to one end. When mixed into a sample, this RNA gloms onto the human DNA and only the human DNA. Scientists then pour in tiny metallic beads that, here's the key, also have chemical Velcro attached, locking the beads and the RNA DNA strips together. Finally, a magnet holds onto the beads as the unvelcroed microbial DNA washes away, leaving behind pristine samples. While this technique is expensive, developing the RNA bait can cost 300,000. New methods promise to drop that price to roughly $50. Okay, so the last paragraph gets into the details of this technique called RNA bait, which is used to decontaminate ancient DNA. And it talks about the details of that technique and how, while it is expensive, new methods promise that its price will drop. So if you look at the two passages and if you try and articulate the relationship between the two passages, you can see that the first passage introduces the problem of contaminated ancient DNA, talks about the rules which have been assembled to decontaminate DNA, discusses the scope of those rules, the limitations of those rules, and finally advises greater care in sampling ancient DNA so that the need for decontamination doesn't arise. I think the second passage also talks about the problems with decontaminating DNA, but takes a more optimistic view of these technologies than the first passage does. The second passage is hopeful that these technologies will become less expensive as the RNA-based technology is talked about. And it also kind of has the tone of being hopeful that these technologies can be the future of DNA decontamination. Okay, so let's look at 47. It can most reasonably be inferred from passage 2 that DNA shredded by enzymes is most beneficial to scientists when the degree of shredding is high, protected from further destruction after cells die, used as material to support living functions in healthy cells, preserved by water and oxygen in healthy cells. So the concept of shredding comes from here, right? Where is the, yeah, DNA disappears after cells die for a few reasons. All healthy cells have enzymes that shred DNA to recycle it. And unfortunately, those enzymes keep shredding after death. So these enzymes keep shredding after death. So the DNA shredded by enzymes is protected from further destruction after cells die. That's not true, right? Because the enzymes keep shredding. So B is out. Water and oxygen also react with DNA's backbone and degrade it further. I would say these processes reduce DNA to confetti. So what is most beneficial? So the degree of shredding, if it is high, it is not beneficial to scientists. So A is out. DNA shredded by enzymes is used as material to support living functions in healthy cells. Yeah, to recycle it, right? So C is a good answer. It is oxygen and water are referred in the context of contamination, so that's not correct. So DNA shredded by enzymes is used as material to support living functions. That comes from here. All healthy cells have enzymes that shred DNA to recycle it, right? So that's 56 to 57. That's the evidence. Yes, 56 to 58. That shred DNA to recycle it. And unfortunately, those enzymes keep right on shredding after death. Right. So, yeah, so it is option A. Okay. Okay, so that was the first part of that line is evidence for option C. When the author of passage 2 says that scientists think they can push that back, he most likely means that scientists believe that line 75 to 76. Scientists think they can push that back to 1 million years, which means that the oldest recovered genome so far came from a horse's leg bone, but scientists think that they can push that back. So they feel that the oldest specimen age can be pushed back to 1 million years, right? Certain limitation might become more pronounced, particular boundary might be extended, scientific theory might be refuted, chronological sequence might be restructured. So a particular boundary might be extended. So the oldest so far is 700,000 years, and that might extend to 1 million years based on this new information that's available. You're not resequencing the chronological order. So D is out. Yeah. Regarding scientific research involving eDNA, both passages imply that even the smallest quantities of recovered eDNA contain traces of contamination. The analysis of eDNA has led to a greater understanding of RNA. I don't think the first passage talks about RNA. Evaluating traces of eDNA is generally problematic. Yes, this is something they both agree with. The usefulness of eDNA analysis is somewhat exaggerated. No, I don't think they call its usefulness exaggerated. The first person has a cautious approach to analysis and being able to decontaminate eDNA, but the usefulness is not exaggerated. Yeah, so I think evaluating traces is problematic. This is true because the first paragraph of the second passage talks about that. And right, and the second passage is kind of, throughout talking about the nature of problem with evaluating aDNA, right? Okay, so I'll go with C. This is something they both agree with. Given the discussion of the RNA-based technique in the last paragraph of passage 2, would applying that technique be useful in the circumstances considered in passage 1, lines 27 to 31? Okay, this latter problem is particularly pertinent if the target DNA and the contaminant DNA are from the same species, as is often the case when research is conducted on ancient human material. Okay, and what are we doing with RNA bait? If searching for human DNA, researchers would prepare the bait by manufacturing millions of strips of human RNA, albeit with one modification. When mixed into a sample, this RNA gloms onto the human DNA and only the human DNA. The scientists then pour in tiny metallic beads that also have chemical Velcro attached, locking the beads into the strips together. Pristine samples. Yeah, but that pristine sample is when you're trying to get human from non-human, right? Because here you are looking at contamination by fungi and bacteria. But that problem is not going to, that solution is not going to solve this problem, because here you are looking at finding contaminant DNA in, which is human, in target DNA, which is also human. So this technique wouldn't work. So it's a no answer because the RNA-based technique is used to isolate DNA of one species from contaminant DNA of other species, right? Human DNA from contaminant DNA of bacteria and fungi. But that's not what the problem said. So A is the best answer. Which choice best states the relationship between the two passages? Both passages consider a proposed approach for solving a problem, but passage 2 argues more forcefully against implementing that approach. Both passages examine the difficulties in a line of research, but passage 2 gives more detail about how scientists are addressing those difficulties. Both passages present hypotheses that are untested, but passage 2 provides more evidence in support of its hypothesis. Both passages raise concerns about the validity of research findings, but passage 2 more effectively articulates the logic underlying the concerns it expresses. Okay, so I don't like A and D because, yeah, the first part of A is fine, that both passages consider a proposed approach, which is, DNA decontamination. But passage 2 argues more forcefully for that approach, not against, right? Passage 2 is a little more optimistic, so A is out. In D, both passages raise concerns about the validity of research findings. I'm not sure that's correct because that's not the point of the passages, right? The point of the passages is to look at the scope of DNA decontamination. Okay, B is a good answer. Both passages examine the difficulties in a line of research, but passage 2 gives more detail, right? So we know that passage 2 actually goes into the RNA bait strategy, etc. So I like B. B is a good option. Both passages present hypotheses that are untested. I don't think they do that, right? It's not about hypothesis testing. It's about looking at DNA decontamination. So I'll go with B. Okay, so let's grade the answers from 43. Okay, BADD, BADD, 47, CABC, CABC, and AB. AB. Okay, so we got all correct. Right, so this finishes the solution series for the May 2021 US SAT. As I said, I have already uploaded solutions to the math sections and the writing sections, and before this video for the first 4 reading passages. So I'll put all of these in a playlist so you can access them together. Hope this was useful. If it was, hit like and subscribe to the channel. I'll see you guys in the solution series for the May 2021 International SAT. Until then, goodbye.